Thank you, Federico. So good afternoon, everyone, and welcome to this webinar, Better Calves in Better Farms. And this is a webinar, part of a webinar series of the EAAP. I am Angela Costa from the University of Bologna, and today I represent the Cattle Commission. And I will share this webinar with Laura Boyle from Teagasc. And Laura, please.
Yes, good afternoon, everyone. Great opportunity to talk about calves this afternoon. I'm Laura Boyle. I'm president of the Health and Welfare Commission of the EAAP, and I work with Teagasc, based in Moore Park Research Centre in County Cork in Ireland. Are you moving on to the next slide, Angela? And
yeah. So I take the opportunity to invite all of you to attend the next EAAP meeting in Lyon, where we will have this session that is a joint session with the Genetics Commission, and that is entitled Better Calves for Better Farms, How the Young Stock Can Be the Key of the Success. So we will have different type of presentation, a multi-topic session, and also an invited talk. So looking forward to see you there in Lyon.
Great. So this is the lineup for today. We have three great speakers. We're very excited about to hear their talks. Angela and I felt that young stock in general get neglected, and we're very happy to spend the afternoon talking about calves. So I'm particularly happy to introduce the first speaker, Dr. John Mee, who's actually a colleague of mine in Teagasc. And John is going to give you a little bit of background about his own career and biography when he speaks. And he's going to talk to us about poor dairy calf health, why and what can we do about it. So thanks, John.
Thanks, Laura. Super. Thank you, Laura. I'll start by thanking the EAAP for this invite to speak to animal scientists across Europe and the world today. It's a great opportunity. My talk is going to be quite broad. I'm going to discuss three topics. The first one is how divorced research is often from reality. I'm going to introduce a topic we tend not to discuss, which is farm blindness. And I'm going to end on a more positive note by giving an example of a national calf health program that may be suitable for rollout in other countries internationally based on our own experience here in Ireland. So that's what my presentation hopes to cover over the next 25 minutes. So briefly, my background, I'm a vet and I work in our National Dairy Research Centre. I've spent a lot of time working across dairy, beef and sheep, both nationally and internationally, and on topics as listed there from health to repro, welfare and production. I also hold some university appointments. And I supervise postgrads across topics, primarily in dairy and beef, but also some sheep research also. And I'm always interested in international collaborations, and this is obviously a great opportunity to connect with scientists internationally, primarily in Europe, but not exclusively. So let's start with poor dairy calf health. Why is there still poor calf health, and what can we do about it? That's what I'm going to discuss. So the spotlight is on calf health. And I'm really asked the question, after all these years of research, and I've participated in this research, there's something wrong here if calf health is poor now. That's really the contentious question I'm posing. So really poor dairy calf health? But we know all the risk factors for poor calf health. So you can see in the slide, you've got an extremely dirty calving pen. So we know that's a risk factor for morbidity in the newborn calf. We know the causes of that morbidity. For example, we've looked at all the viruses and bacteria and protozoa that cause these problems. We've advanced diagnostics and pharmaceuticals available to farmers and veterinarians in the field. So we should not have poor calf immunity, high calf morbidity, and high calf mortality today in 2023. But let's look at the reality. So successive passive transfer immunoglobulin is a cornerstone of good young stock management. We probably all agree with that at a research level. But let's look at the reality of what happens in the field. So I had the opportunity recently to review the international literature, whereby I chose recent studies and large-scale studies across the world. And as you can see from the example here, the US does quite well. Only approximately 10% of the calves have FBT on a very recent nationally representative survey. We go right out to over 40% on a recent Canadian study. And Ireland doesn't fare particularly well, approximately 40% of calves with FBT. So despite all we know about this, this is the norm internationally. Let's look at calf morbidity. So I had a really good opportunity with a PhD student recently, Mary Claire McCarthy. We did an extensive herd and calf level study. We looked at over 6,500 calves in 120 herds. I'm just going to focus here. There's both herd and calf level data, but just at the herd level data. So if you can see on the third line of the slide, in 89% of farms, we could find at least one calf with calf diarrhea. And if we follow across the slide, we could find that 40% of farms had a calf with pneumonia, 37% had a calf with high temperature, 100% of farms had a calf with an enlarged navel, and over half of farms had navel ill. So these morbidities are extremely common at farm level, less common, obviously, at calf level. So the ultimate end of all this is calf mortality. So we should expect this to be low, given all the research we've done. No, not so. So at one extreme, we have extremely low calf mortality in a slightly dated study from Switzerland. And you can see the definitions of calf mortality here. Essentially, I'm talking about from roughly a day of age up to 6 months, the pre and post weaned calf. But at the extreme, this can extend out to over 20%, but let's say on average, it's about 5%. And it hasn't changed greatly over time. And these are fairly large scale studies. So again, hugely disappointing. So why do we still have this poor calf health? One thing that's happened is we've deprioritized good calf health while we've reprioritized other issues. So the relative prioritization has changed over time. I give the example in Ireland over the last 10 years, if you're a dairy farmer, your number one priority was improving the fertility of your dairy herd, not improving calf health. Not that you didn't do it, but relatively speaking, it wasn't your priority. We also have an issue of hazard perception discord between the farmer and their stakeholders, particularly their veterinarians, but also their advisors. So there is a gap between the expert and the farmer, and that affects prioritization. We do also have poor young stock management. That's failure to implement recommended procedures. And I gave the example in failure of passive transfer, and I'll show some other examples shortly. And this issue of farm blindness, which is the misperception by the farmer of their management as being the norm when they don't know what the norm is. So in order to research calf health in Ireland and try to improve it, we carried out a Delphi study back in 2010. These data are dated, but I will update them. We asked experts and farmers, what are the most important conditions we should work on to improve in Ireland? And if we look at diseases in young calves, we can see that experts consider them 5th in ranking out of 13, dairy farmers 3rd in ranking, and beef farmers, as expected, because the calf is more important, 2nd in ranking. But number 1 in ranking then was infertility. So you can see why in an environment like this, calf health is not going to get the priority when it's not ranked as being that important by farmers or stakeholders. That has changed quite a lot in the last 10 years. This is the hazard perception discord I mentioned. This is a nice Dutch study which shows rankings of vets and farmers here. So take the example of diarrhea in the older calf. It was ranked number 1 by the vets, but only 5th by farmers. We look at calf pneumonia, in this case ranked 5th by vets, but number 2 by farmers. So the priorities of the farmers and the vet don't often align. And if they don't, then prioritization is impossible because one is speaking apples and the other is speaking oranges. So let's look at poor young stock management. There is evidence of procedural failings and failures to implement recommended management practice. These are the protective factors. And I'm going to give examples here. I'm going to quote a line that we probably all agree with. And then I'm going to look at the evidence to see, well, does that actually happen in practice? And I'm using 2 simple examples here, umbilical care and colostrum management. So we probably all agree with this, that it's recommended that naval care should be carried out in a newborn calf to prevent umbilical infections. And I know there are contrary data in this field, but as a general principle, let's accept it. Now let's look at what happens in the field. So again, this is an international survey of what's happening at herd level, recent data. And we see that these vary between 90% of herds in the Czech Republic do use naval disinfection, And in contrast, 90% of herds in India don't, and wide variation internationally. So it's not the norm to use umbilical antisepsis. It's the norm to have wide variation in what people do, despite us all agreeing with this statement. If we look at measurement of colostrum quality, we'd say that measurement of quality of colostrum fed, particularly the first feed, is recommended. This is good practice. Do we do it? At best, up to 56% of farmers in the Czech Republic don't do it. Right up to 90% in Brazil don't do it. And Ireland ranks in between. So again, something we recommend should be done, doesn't really happen in practice. Feeding pooled colostrum. It's a route of pathogen transmission for the newborn calf, and it can dilute first colostrums, so not a recommended procedure. What happens? In the US, only 20% of farmers do it, so it's quite good in their relatively recent data, right out to 80% in Norway. And again, Ireland ranks somewhere in between here. So even though we may say this really isn't something you should be doing, no, farmers still do it. Measurement of failure of passive transfer. The same here, when we look at the variation, 86% of herds in Canada, or farmers in Canada, don't do it, right out to almost 100% in Brazil. I don't have Irish data, but we'd be somewhere in the same range. So measuring failure of passive transfer on farms as a routine procedure is not carried out. So how does this clash with reality? Well, this is an example of a recent case study of 600 cow dairy herd, where they lost 15 calves. 15 calves died in a very short period of time. And the signs were non-specific, indicating perhaps an enteric disorder, but perhaps something else. There was queries over the iron level in the water. There may be toxins in the cow's feed. The milk replacer may be poor quality. Maybe there's a very virulent bacterium or parasite involved. A lot of calves had died. So the veterinarian was involved, the laboratories in Ireland were involved, and it came to the end of the study that the problem was E. coli septicemia. So an infection with a relatively common pathogen on a farm. Why had this occurred? So thorough investigation revealed, one, it occurred during peak calving. So that represents about 85% of the farmers in Ireland. That's not unusual. The farmer was pooling colostrum. Half of farmers in Ireland pooled colostrum. He stored it at ambient temperature. I guess over 90% of farmers do that. He didn't test the colostrum quality. We know that I've just shown you 84% of farmers do that. He didn't test the calves for failure of passive transfer. I guess over 90% of farmers don't do that. He didn't use any umbilical antisepsis. That is slightly unusual in Ireland. And there's widespread AMR, antimicrobial resistance, in the isolates detected on the farm. But the farmer did stomach tube colostrum and did vaccinate, which is unusual in Ireland. Only 28% of farmers do this. So this is an example of what could be described as a farmer doing typical management procedures, and at some point, a constellation of things go wrong at the same time, and there's an outbreak of deaths. That's the reality. Despite all we know about the research, this is what happens on the farm. So why do these problems still exist? So I'm going to discuss briefly farm blindness, and I'll give you a definition of it there, that it's a misperception that what farmers see every day on their own farm is normal. It's the same as every other farm, when it's not. They're creating a new normal. And we see evidence of this with failure to implement these basic practices and failure to recognize, in this case, high calf morbidity or mortality. And this is a very good example from a recent Dutch study where farmers had high calf mortality. They were investigated and asked why they had this high mortality. And in 46% of the farmers, they weren't aware of their losses, despite the fact that they had high mortality. So they just were blind to the losses on the farm. So one of the reasons this occurs is that farmers don't recognize the problem. They don't record abortions. This classically occurs, particularly for early abortions. They're not recorded, so they don't appear to have a problem with abortions. The same occurs with stillbirth. I've already mentioned the aberrant has a perception with calf diarrhea. The veterinarian may consider it a problem, but farmers may not consider it a problem. They may recognize the problem, but be blind to it. In the classical case is lameness. 20% of their cows are lame, but they're desensitized to it because lameness occurs at 20% every year. So it's normal. We may also have the issue of change blindness, where something bad slowly becomes normal. So for example, drift in vaccination dates, they were correct 10 years ago. Now they've drifted to the wrong time of the year, but that drift has not been perceived as occurring. And pluralistic ignorance is a major issue. Things like body condition scoring. not realizing your cows are fat or thin because you assess them yourself. So an example of this would be the farmer who's ignorant to what other farmers are doing in young stock management and how their calves are performing. This is a direct quote from a recent paper, I think it's a JDS paper. The farmer says, well, to be honest with you, I probably never talk to anybody about calves. So if you don't, then you don't know what other farmers are doing. And I've already mentioned this discordance between the veterinarian and the farmer and the information asymmetry between the two. Okay, so that's painting a rather dismal picture of calf health. So the next question is, what can we do about it? We need to reprioritize, and that's occurring here in Ireland now. There's a recent emphasis on calf welfare specifically, and a refocus on AMR rather than the problem that the treatments used for the problem. We need to improve young stock management independent of problems, just basic management. So I'm going to discuss a national dissemination program for this, on the communication of best practice rather than problem practice. We need to address farm blindness, which is creating awareness so the farmer isn't blind anymore, providing benchmarks which they can use that are relevant to their production system. And agri-tech does have a role here. PLF does have a role. So I mentioned that 10 years ago we did a Delphi survey in Ireland. We reissued this survey in 2021. Again, dairy and suckler farmers and their service providers and the farmers here, and you can see the ranking of priorities. And now the ranking on AMR has changed totally. It's now a major issue of priority, both amongst farmers, whether they're dairy or suckler. Calf welfare ranks reasonably high, but perhaps disappointingly slightly lower amongst the farmers than amongst the professional service providers. But remember, farmers have more things to worry about than just calf welfare. It's relative prioritization for them. So one of the things we've done in Ireland, and we're not unique in this, is set out a calf health campaign, providing a national or quasi-national infrastructure. And these are examples of logos of this internationally. Some are public good, some are private good. The example in Ireland is our animal health organisation, Animal Health Ireland, AHI, and it's called the Calf Care Campaign. It's been running in Ireland since 2010, and there are other examples internationally here. The national objective, certainly within Ireland, is not just to identify and record calf health issues, but to improve them. And in our case, in a rapidly changing farming environment, like since prior to quota to today, we've had a 64% increase in our milk output. Massive change, unique within the European farming context. So that means there's more calves every year in our farms, extremely challenging for calf health. So campaigns will be run every year. An example might be using something like how to provide better colostrum management, and we use a simple mnemonic, colostrum 1, 2, 3, or knowing the stocking rate inside in your head, know your numbers. And using national benchmarks, and we're looking to have our ICBF, our National Breeding Organization, that provides data on these metrics for us. And essentially what we're attempting to do is respond at scale, rather than just address a farmer with the problem, to provide the underpinning across the industry of better calf health management. So the basis of this is, one, that anything that's said is evidence-based. A classic example would be that prior to this campaign in Ireland, we would have stopped feeding milk to diarrhea calves. Now we advise continued feeding of milk to diarrhea calves as an example. The messaging is consensus-led, so we all agree on the message. I gave the example of colostrum 1, 2, 3. So first colostrum should be only the first feed. It should be fed within at least 2 hours and should be at least 3 litres. A dissemination infrastructure that involves both Teagasc, this is our National Agricultural Research, Advice and Education Organisation, in combination with AHI and critically in combination with our cooperative organisations. All communications in farmer language, it's not professional jargon. The strong peer learning using discussion groups on farms or in meetings, and providing resources to farmers, whether that's leaflets or videos, so that they have information that they can get easily. I mentioned the issue of farm blindness. Some of the ways of creating awareness is having a fresh pair of eyes visit the farm. The vet performs that role, or the consultant, or the agricultural advisor. And these national level campaigns also perform this role of creating awareness of issues. Then you need to provide benchmarks against which the farmer can compare their performance. So these are the iceberg indicators. That could be the proportion of calves that die within 28 days of being born. These criteria can either be norm reference using the population at risk, or they could be criterion reference, whichever is available. And these are either best They're nationally or regionally sourced, not international data. And we know from the literature that providing benchmarks alone motivates change practices and provides better outcomes. And I mentioned there is a role for PLFs. So some of this tech that is increasingly being used on dairy cows is now being used on calves. And that does provide, to a degree, farmer independent monitoring. So even if the farmer isn't aware of the problem, the technology alerts them to the fact that there is a problem on their farm. So a lot of what I've said prior has been about the problems that have occurred, but our animal health organization went back and resurveyed our farmers in 2020 and asked for their sentiment about how calf diseases, what had happened in the period between the start of that campaign and now. And you can see that the majority of both dairy and suckler farmers and their service providers believe that in that time, calf health is better, either somewhat or much better. So the sentiment in the industry is that this campaign nationally has been a success. So really what I'm advocating here is not accepting the norm, advocating change and change at scale. So for those of you that are interested in reading further, and I've obviously only cited our own papers there, but there's lots of international literature on this area about both problems in calf health and what we can do to affect change, which is really what I'm interested in, not just identifying the problems, but doing something about it. And in a rather shameless plug, I've recently co-edited a book with colleagues that deals with many of these issues, and it's available for those of you that are interested in it. So I think I've taken up enough of your time, and I'm now happy to take any questions. Thank you very much.
Thank you very much, John. Yes, thanks a million. I think there might be a question from the audience there. So I can read it out. Which do you consider bigger issue, the management problems I said before, or the circumstances in which the calves are being held? For example, the temperature, older animals in the same barns, etc. Which is a management problem.
Yeah, of course. The management is more important than the facility, because we find that we will have calves with health issues in both good and bad facilities. And the differentiating factor is the quality of the management. So that's why, to me, the critical factor is the person who manages the facilities, not the facilities per se. They're both important, but I put the balance towards the management.
Okay, yeah, that makes sense. John, I was just wondering, would you comment on something you said earlier? And it's something I think we may have discussed previously, but I was interested in that farm that you characterized that had the high calf mortality, and that they were unusual and that they were vaccinating, and also that they had quite a high rate of antimicrobial resistance, if I'm right. And could you comment on the fact that some farmers tend to rely too heavily on this silver bullet issue? I suppose we see it across animal production industries, that the other issues are so complex, you know, in tackling them, that there can be this other maybe slight problem that could be contributing to all the issues that you laid out, this reliance on kind of a silver bullet to managing calf health when it's multifactorial and needs to be broader.
Yeah, it's a good point, Laura. So vaccination by farmers is often seen as, well, I can't have the problem because I vaccinate against it. Mm. But in this particular case, the vaccine the vaccine does provide protection against E. coli, but only specific strains and not the strains that cause the problem on the farm. So that granular knowledge is not available to the farmer. Their assumption is that if I vaccinate against E. coli, then E. coli can't cause a problem on my farm, which is not correct. So that's a failure of knowledge transfer to the farmer. That is all our fault, that we don't get that level of knowledge there. What it means is that if you vaccinate, you're vaccinating the cow, the protection is in the colostrum, so you need really good colostrum management, and you need to realize that alone does not prevent calf diarrhea due to E. coli. There's lots of other, as you mentioned, complex issues that are much more important than the vaccination itself, as it proved in this case. The farmer was vaccinating, thought he was doing a very good job in doing so, but there was much more going on the farm than just vaccination. That's a failure of knowledge transfer.
But could there's also be a failure of, well, I can neglect all those issues because I'm being covered by the medication or the microbials or whatever, the antimicrobials or whatever?
No, I wouldn't say it's the farmer purposefully saying, no, I won't do this because I do that. It is perhaps relying too much on the vaccination and perhaps not placing the emphasis on the other issues. It's not that they believe that just because they vaccinate, they don't need to do the other things. It's perhaps they deprioritize them because they vaccinate. So again, okay, it's a knowledge transfer to say, if you do one, you also need to do the other. It's not one or the other. It's not that simple. So yeah, your silver bullet versus complex is a very good analogy. It is always complex. It's never a single bullet, ever.
Great. Angela, you might remind me or tell me when we have to get started with Juan's talk, but because there are
no, we have time, but yeah, we have also a lot of questions in the
yes,
chat box.
Exactly. So yeah. Do you want
to start to read?
Yeah, you can call out the next one too.
Yeah. So, John, a question from Diana. How do you think there is room to improve cow's health through the revision of the European law on welfare that will happen this year in 2023?
Yeah, I suppose you have the EU law that needs to transpose into national legislation, and then it needs to be communicated through, in our case, our Department of Agriculture, and we have active ongoing discussions with our Department of Agriculture regarding the timing of that process, the phasing in of that legislation, and farmers basically need to be communicated well in advance of any change that will materially affect their facilities and their management now and their future management of facilities well in advance of it becoming law, so that they can adapt to it. And then communication at ground level of the implications for farmers if space allowances change, if age, transport changes, any of these critical legal changes that occur. It's all about communication in advance to farmers and to the service providers, to the agricultural advisors and to the veterinarians on the ground who have to pass this information through to farmers, that they're up to date in advance of the effect of the legislation coming into law in the country. So that to me is critical well before the legislation is implemented.
Yeah. And we also have another question from Guyadieu. Did you work on the different way to deliver advices on the subject? Are farmers blind because of the difference of vision they have with veterinarians? Who are their advisors? Is there something to do not only on technical messages, but also on the way to deliver it?
Yeah, that's a very good question. So the data show that the perception of the farmer and the veterinarian can vary quite a lot in what's important on the farm. And independent of that, there are lots of data showing that the way in which the veterinarian or the advisor communicates that advice to the farmer impacts how much of that advice is taken up by the farmer. And the two issues are then interlinked. So there's two issues. One, the farmer has a different perception to the veterinarian of what's important. And two, how the veterinarian impacts that advice impacts whether the farmer will take up that advice or not. Yes, they're both important, both part of the solution to farm blindness.
So, Laura, it's up to you. Which question would you like to
yeah, there is another question. There's actually one up further up towards the top, and it's about self-controlling systems such as private assurance schemes. Would you support the monitoring, reporting, and improvement of general health and welfare through QA, through quality assurance schemes, I suppose?
Yeah, I mean, QA schemes have got a bad name internationally because often they're tick box exercises. They don't have animal-based measurements in them, and they're not useful for animal welfare or animal health. But QA schemes that have realistic metrics for farmers to achieve, so benchmarkers that they can actually achieve within their industry, focus on the animals rather than the facilities, not a stick, but a carrot, so that the farmer can see the benefit of applying these schemes on their farm. If you take all of those approaches, you'll improve the reputation of these schemes and likely improve compliance with them as well. In principle, I totally agree with them, because we can't go on with poor quality animal health and welfare without doing something about it. And QA schemes are one way to attempt to improve it, but they're only one way.
Are we still okay, Angela, for one more question? For time,
yeah, we have time. Yeah, we have time until
the program open in front of me. But
no,
there's
we have 8 minutes left.
Okay,
so.
okay, great. But because there's a nice question from Frank Titans from, I think it's Frank from Ilvo, if I'm right. And he's asking a question which I would have liked to ask as well. So I'm glad he asked it, because it's about the value of the calf and the role that plays in their welfare. And what Frank is basically saying is that, you know, the core of the well, a big part of the problem, and I think he's could be right, is this difference in economic value between calves that are produced from milk and versus meat. And could you comment on that, please?
Okay, so you can divide it into the enterprise first. So the beef calf is perceived by the beef farmers more important than the dairy calf is by the dairy farmer. That's why beef farmers rank calf health issues higher than dairy farmers, because the dairy farmer primarily produces milk as the economic output. The beef farmer produces their calf as their output. So there's an immediate divergence there. If we go within the enterprises, then say the dairy farm, we have the divergence between the economically lower value calf on a dairy farm that perhaps might get lesser quality of management and then be more susceptible to mortality and morbidity. But in the case history I showed you there, the farm had calves that were not economically of lower quality. These were top quality calves that were dying. So there was no attempt to provide poor quality management to them. The farmer was doing the best they could to provide the management. They still had problems with morbidity. So we can't hang poor calf welfare and poor calf health just on lower value calves on farms. It is part of the issue, but it doesn't encompass all of the issues. It's just one aspect of it. But I will have to say that increasingly, in industries that are producing more lower value calves, you may then get more lower value care of those calves. That is a risk. That's a genuine risk. But it's not generic to the industry. It may be farm specific, but it is a risk we do need to address. Yes.
But John, the statistics would show for a long time there's a higher rate of mortality in male calves versus female calves across internationally. And I mean, I don't know that it's that biological difference in that males are more susceptible to death. I don't think that explains the magnitude of the difference. So I think that would show that there is a divergence in care, or suggest.
Yeah. I suppose the higher death rate of male calves occurs right from birth. More of them die during birth, which is, you could debate. Farmer at that point may or may not know the sire of the animal, but it does occur right from birth right through. You are right. There is a gender difference. And you are right to say that it's likely that if the male is of lower value, then the likelihood of poorer care is higher. So apart from the gender difference, the management difference is likely to be more important. It's hard to argue against that because the facts are there on the gender-specific death rates. Yeah, it's hard to argue that.
Angela, have you any other questions?
Yeah, there might be an important one, maybe the last one. Do producers have the perception that cows are the future milk-producing cows in the dairy system?
Very much so. And for example, in Ireland, we have latterly taken to much increased use of sex semen. We lag behind some, perhaps the North American countries, but it's increasingly being used in Ireland now. So farmers that are producing calves from sex semen would absolutely see them as the future of the dairy industry. In 2 years' time, they will be producing the calves of the future. We have huge emphasis within our dairy industry on functional genetics. Major focus. All of our top dairy farmers would be really well educated, knowledgeable about the genetics of both production and fertility and health across the sphere of sires they use. So that's a real focus for the future, in that our farmers will be very, very focused on what's happening in 2 years' time in the industry, and genetics obviously has a huge role in that. So it's a yes, very much so.
And
there is, okay, I'm just going to throw this one in, Angela, while you're looking, but it is asking, I think you may have answered it, about the importance of management, but someone is asking about how much of a role the genetics of the calf, sire, or dam affect calf mortality on farm.
Yeah, we accept it now that we have within our EBI, we have health traits in the EBI, so we accept that genetics has a role to play, but we don't want to overplay that role either. We don't want to kind of say that genetics will do it for us and management doesn't matter. Genetic just loads the gun, we pull the trigger. We decide in our management what happens to the genetics that we have on our farm. So we can't either use it as an excuse that we have bad or good management or a get out clause. But it is part of how nationally Ireland Inc. and other countries internationally are going to address these problems. Is they're going to produce more resilient, more robust calves with better health through genetics, a slow cumulative process over time. But the opposite to that is don't address it, and you will be producing calves with greater susceptibility. The problem is that the emphasis within a genetic selection index is 100%. If you put 2% into calf health, you have to take 2% out of some other trait. You can't have 102%. So it then becomes what's economically the most viable thing for the farmer. And if we decide in Ireland that calf health is worth 2% in the index, 2% will go in. And if in some other country they decide it's not economically viable, it won't go in. So that's the reality of applying the theory of genetics to the reality on the farm.
Yeah,
Very
true.
good.
I agree totally. Yeah, economic weights are really, really important when it comes to inclusion of new traits, particularly for traits that are not directly related to the dairy cow, the lactating cows itself. So yeah, sure. But I think that maybe the next speaker will provide a more exhaustive overview of the genetic aspects related to calves, calves' health, and how the calves' genetics interact with the dam genetics, right? So it's a pleasure to have here Juan Cordero from the SLU, from the Swedish University of Agricultural Science. Juan has a PhD that is joint between SLU and the Wageningen University, and he did a great job studying the colostrum and how the quality of colostrum was interacting and correlated with the calf serum antibodies. And so today we will have an overview of your main outcome and findings, Juan. So please, the floor is yours.
Oh, thank you, Angela, for that lovely introduction. As she was mentioning, today I will be presenting what was part of my PhD project, where we take a look at the genetics of antibodies and colostrum, newborn calf serum, as a way to, let's say, lay a hand on trying to tackle FPT a little bit. But of course, this is a much more complex issue, but we just hope to put a little grain of sand, let's say, on this issue. So, well, colostrum is the very first milk produced by the mother shortly after giving birth. This initial milk is quite different from the regular milk that comes later. It has this yellow color and thicker consistency. This very unique appearance is not only visually distinct, but it also indicates all the vital nutrients and components it contains. One key aspect that sets colostrum apart from regular milk, it's a significantly higher protein and fat content. These elevated levels of protein and fats play a crucial role in providing the newborn with the necessary energy and building blocks for rapid growth and development, well, during the first few days of life. But perhaps the most remarkable feature of colostrum is its abundance of antibodies, especially, and other immune components like cytokines and leukocytes. These elements serve to protect the newborn from infections and diseases at a time when the immune system is still quite immature. And the transfer of these maternal antibodies help to establish passive immunity in the offspring, which can be critical for their survival in the early stages. So antibodies in general are classified into isotypes based on their heavy chain structure. We have IgM, for example, which is the first isotype produced, allowing a quick response to a variety of pathogen molecules and other antigens. Upon antigenic stimulation, the B cells, which are the cells producing the antibodies, can switch isotype and convert into plasma cells that produce specific antibodies. For example, we have IgA, which is mainly found in mucosal secretions like saliva, tears, and respiratory and gastrointestinal secretions. Then we have IgG, which provides long-term immunity, neutralizes toxins, and facilitates phagocytosis by immune cells. In bovine colostrum, IgG is the primary immunoglobulin that confers passive immunity to the calf. The way that these antibodies get from the mother to the offspring depends on the species. It can come directly to the neonate in the womb or to the mammary gland during colostrogenesis. Some species, like humans, only get the antibodies in the womb, in utero. Other species can get it from both. But most livestock species, including bovines, get it only from the colostrum, which is making it extremely important for them. This fact has an implication on the colostral antibody composition. For instance, in these two first groups, the main antibody is IgA. But for livestock species, including bovines, IgG is the main colostrum antibody. Another implication comes at the gut level. The first two groups have none to a moderate gut absorption of antibodies, whereas for bovines is quite extensive. Now, serum IgG concentration in the calf is used as an indicator for passive transfer success. A concentration above 10 grams per liter measured between 24 to 48 hours after birth is generally considered as the cutoff for adequate passive transfer in calves. If this doesn't happen, something called failure of passive transfer occurs, where the calf doesn't get enough antibodies. This, as John quite nicely showed, is quite prevalent in Europe, and Sweden is not the exception, with prevalences ranging between 30 to 40%, which is quite high. Animals with failure of passive transfer are have twice the risk of morbidity and death, mostly because of an increased risk of scours and pneumonia. Failure of passive transfer has also been associated to milk yield later in life, and even so with average daily gain, showing that this is something relevant not only during calf age, but also later. So which factors influence passive transfer? Well, there are many. But let's name the timing of first colostrum feeding is quite important, because there's a short window where the calf is quite receptive of these antibodies. And of course, the volume of colostrum consumed. And we can go on of other factors, but let's mention just these two. These are highly dependent on management and have a strong factor effect. But then we have something like the quality of the colostrum, which is defined as level of IgG in the colostrum. If it's above 50, it's considered good quality colostrum. And then we have the calf's unabsorption capacity. And we believe that these two factors have a genetic component that we can breed on. How do we assess passive transfer? Well, we measure colostrum quality and serum IgG. We can use direct methods that measure the antibodies directly, such as ELISA or radial immunodiffusion. We have other indirect methods like Brix percentage, which is a refractometer system index for colostrum, or we can measure serum total protein, for instance, in calf serum, which are the traits that we're going to use here. Then when we talk about antibodies, antibodies in general are highly influenced by the environment, especially the clinical history of the animal. So we wanted to include as well what are called natural antibodies. These antibodies are part of the innate immunity, so they don't require previous exposure of a pathogen or specific antigens to be there. And they are polyreactive and have a low binding affinity. The quality of being polyreactive meaning they can bind to some pathogen-associated molecular patterns, for instance, or autoantigens, which are antigens from the own cell. And this provide them with some homeostatic properties, cleaning cell debris and others. Natural antibodies in dairy cows have been associated to risk of mastitis, lameness, postpartum uterine health, productive life, and negative energy balance. And in chickens, they have been associated to longer survival and even E. coli resistance. So there's some interesting association of these antibodies with general health. So I'm just going to briefly mention general concept of genetics of quantitative traits. Complex traits like antibodies are polygenic, meaning a high number of genes within the genome have a small effect that in the end, the sum of these effects are what define the trait. So we need to analyze these complex traits at the population level in order to account for the environmental effects. When we do quantitative genetic analysis, we look at the heritability, which is, let's say, the measure of how much variation in a trait is due to genetics versus environmental factors. And it ranges from 0 to 1, where higher values indicate greater genetic influence on the trait. The genetic variance has a higher influence on the trait. And this helps us identify traits that can be improved through genetic selection. Then we have genetic correlations, which occur when 2 or more traits are influenced by the same genes or genetic factors. This genetic correlation can be positive if both traits increase together. So if you prefer for a higher level of antibodies, other traits will also go higher, or negative, say one trait increases while the other one decreases. And this is quite important when you're considering potential trade-offs between traits during breeding decisions. So when we look at what we know about genetics of antibodies, colostrum IgG content and bricks, well, some previous studies have found moderate heritabilities, meaning that there is indeed some genetic component here that we can look into. For calf serum IgG and serum total protein, some studies have found haplotypes for bovine neonatal Fc receptor, which is related to IgG uptake in the intestine. But the truth is that little is known about the genetics, the genetic background of these traits. So in this study, we aim to explore the genetics of antibody content in colostrum and newborn calf serum to investigate if the occurrence of FPT can be reduced through selection. So in our study, we did some sampling from January 2015 to April 2017. We focused on 3 farms in 3 regions in Sweden, and we included 2 breeds, mainly Swedish Red and Holstein, Swedish Holstein, and some crossbreed of these 2 breeds. We had a little over 2,000 samples split between cows and calves. For the cows, we sampled colostrum. We took the first milk in colostrum. That's very important. And since we did this over several years, we had more samples than cows through different lactations throughout the experiment. And for calves, we took serum, mostly from 1 to 12 days after birth, but over 90% of the samples are between 1 to 7 days. We made sure to record, to match the calf serum information also with the cow's colostrum, the mother, let's say, of the calf. We also included some health information. We had 2 groups of health information from 0 to 3 months of age to evaluate, let's say, the immediate effects of the antibodies, and from 8 to 36 months of age to look a little bit later in life. We tried to look into the genetics of milk production related to the colostrum. Yes, and then we looked at the milk production of the calves later in life to see if there was any relationship between these. So for the phenotypes in serum, we took serum total protein as an estimate of IgG, total IgG, and some natural antibodies, but I'll go into detail about these phenotypes. We also took information like weight at birth, first meal, the time that after birth that the calf was fed, how much was it fed, and of course the colostrum quality information, and some health traits, as I mentioned before. For the cows, regarding colostrum, we analyzed Brix percentage as an IgG estimate, total IgG, natural antibodies, and we took record of the time from calving to sampling, which is a very important factor here. So as far as genetic parameters go regarding colostrum, to analyze this, we used a linear mix model. We accounted for some fixed effects that we deemed relevant, including calving to colostrum sampling time, as I mentioned before, parity, breed, and herd year season of calving and sample storage plate, and random effects. We included an additive genetic effect and a permanent genetic effect. In the end, we ended up with these heritabilities, which are consistently moderate, I would say, across all the traits. And very interestingly, the trait that we were hoping to use as an indicator trait was quite good in Brix with 0.31. And then we have the IgG traits, both the total IgG and the natural antibody traits, with some positive, well, significant heritabilities. And for the rest of the natural antibodies, we also had some moderate heritabilities. So regarding genetic correlations, we wanted to know, of course, if these traits were correlated with each other. Interestingly, Brix had a significant positive genetic correlations with all the traits. When it comes to total IgG, had a correlation of 0.68 with a standard error of 0.14, which is quite high, quite good. And with natural antibodies, also some pretty good correlations, 0.49 to 0.73. When it comes to only natural antibodies, IgA and IgM were highly correlated within each other. And something similar happened between the IgG natural antibodies with 0.76 genetic correlations. Now, relating colostrum antibody levels with milk production in the same lactation, let's say, we did not find any significant genetic correlations, but the values had high standard errors, even though it seems that at least the genetic correlations may not be strong, which is a good thing, I would say. We really do not want these values to be correlated with production, interfere, let's say, with any production breeding schemes. So then we try to use to correlate colostrum antibody levels with calf serum levels as a predictor of calf serum traits. We tried to use the cow genetic component for the colostrum traits with the calf traits using the calf genetic component, but the statistical power was not enough for this. So instead, we used cow genetics for both, and we indeed had some positive, at least, correlation, but the standard error was quite high. And then we had some significant genetic correlations between colostrum, calf serum, IgA, and IgM, natural antibodies. So yeah, definitely to try to pull this, we need a larger sample. All right, now going into the genetic parameters of calf serum, we also used a linear mixed model, and we included some fixed effects, including birth weight and the absolute amount of first colostrum given. We multiply the concentration of the colostrum times the volume given to get an absolute, let's say, value of the colostrum given. I think this is quite the most important factor to account for here, in order to not, let's say, accidentally include the mom's colostrum genetics accidentally, let's say, into this. So then we included the time from birth to blood sampling, which is also critical, that the calf breed, herd year, season of birth, and the sample storage plate, and some random effects, of course, the additive genetic effect and the maternal environmental effect. Here we wanted to include the maternal environment effect, account for non-genetic influences of the mother and offspring antibodies. So as far as heritabilities go, we had a bit of different results compared to colostrum heritabilities. Sadly here, the serum total protein, which we were hoping to use as an indicator trait, the heritability was not significant, meaning that the genetic component here we could not have, was not statistically different from zero, let's say. But then interestingly, we found that the IgG traits, including total IgG or serum IgG and KLH and MDP, the natural antibodies for IgG, had a consistently similar heritability, which is quite moderate and quite good, usable. And then for the rest of the natural antibodies, IgA and IgM, we had some quite high heritabilities, surprisingly. Then one of the most interesting parts of the study was the maternal effect, because we had consistently high, or what we would consider consistently high values of maternal effects here. And why is it surprising? It's surprising because we are looking at non-genetic influences of the mother on the calf serum traits, even though, as I said before, we accounted for the amount of colostrum given. So, and also these calves are separated from the calf immediately after birth. So there's no prenatal care here to impact their development. So there's something else in the colostrum or something going on in the womb that may ultimately affect the calf serum, but we don't know what. Genetic correlations between calf serum traits. SDP had no significant genetic correlations, but that's not a surprise. When it comes to serum IgG, interestingly, it was only correlated to IgA and IgM slightly. Perhaps this means that there is no specific mechanisms for isotype uptake related to the uptake by the calf. So now correlations between calf serum and health later in life. When we looked at young calves from 0 to 3 months, we only found let's say, borderline significant association with MDP IgG, and it was a negative association, which is good, meaning that the higher the MDP IgG value, the lower the chance for the calf to be sick. Bear in mind here that we are looking at the genetic variance part of this. Obviously, if the calf has good antibodies, it's going to be protected against disease. But what we're looking at here is like the genetic capacity of the calf to have a higher level of this means that it's almost significant for this. But to be fair, for this part, we really needed more animals, and we had a big, let's say, problem, quote unquote problem, because the calves were too healthy. So we didn't have enough sick animals. And well, for heifers, we did not find any significant genetic correlations. And we looked at milk production later in life. We found some interesting genetic correlations with the lactation average somatic cell score. Total IgG was in total serum IgG had a favorable correlation of -0.62, meaning that the higher the antibody level, the lower somatic cell score or later during the first milking. Something similar was observed for IgM, IgA natural antibodies. So for general conclusions, antibody uptake traits of newborn calves have moderate heritabilities, and selective breeding could be used to reduce FPT. The maternal contribution to calf serum antibodies is more than just the colostrum antibodies. And calf serum traits and health from 0 to 3 months of age had a negative tendency, which is favorable in genetic correlations, but these were not significant. Colostrum traits from the cow may potentially be used to predict the calf's ability to uptake colostrum. BRICS is genetically correlated with total IgG and natural antibodies, so it can be used as an on-farm indicator trait to select for better colostrum quality. And, well, a larger sample is needed to establish clearer correlations to production health and other traits. So thank you very much. I think I'm a little over the time limit. That's it on my side.
Thank you, Juan. Thank you for your brilliant overview. That's really impressive and also important to see how colostrum trait can be heritable. I mean, it's nice, but at the same time, as John said, we cannot use this genetic potential as an excuse to avoid
Absolutely.
management to be improved,
Absolutely.
right?
Definitely.
So we have some question maybe in the chat box. So from Essie, is this study available for further reading somewhere?
Yes, of course. I'll send you the link.
I think you have your PhD thesis, there's some articles as well, right?
Yes.
Yeah, so for sure, and we can maybe write in the chat box the link of your thesis or your article. And one question, maybe you said, Juan, that there is no selective uptake, let's say, in the gut, in the intestine of the cows, right? So it seems that IgG are absorbed equally as IgM and IgA. And would you like to comment a little bit more on this and discuss a little bit more on this? What's your opinion about it?
So at least what has been studied from a molecular and immunological point of view was the FC receptor in the intestine of the calves, which took the antibodies from the intestine, which is where the uptake happens in the small intestine, and then takes it to the bloodstream. But there seems also that through tight junctions, larger molecules and non-specific molecules like IgM and IgA can pass directly to the bloodstream of the calf. So there may be some IgG molecules that also go through that way, and others that go through the FC receptor way. So that probably might create some sort of a genetic correlation of this. But the thing is, like, the studies related to the biology of these are not so complete, or let's say they don't dive too much into this. So the way that these antibodies get uptaken, aside from the FC receptor, is just a big question mark. We can only really speculate on how they are uptaken. We do know, well, I can probably give a little bit of information from the GWAS, that we found that some genes related to saliva and gastric acid production are related to the uptake. So we know that the way that the IgG binds to the FC receptor in the gut is highly dependent on pH. So this may be one important component of this that at least I couldn't find mentioned anywhere else. And we know that the gut of the calf in this first hours with their highly receptive IgG, the gut is quite alkaline. It's not acid yet. So maybe there's something there.
Okay. And do you think that the colostrum quality should be referred to the immunoglobulins concentration merely, or are there any other components in colostrum that might be important and fundamental in the occurrence of the failure of passive transfer?
Well, if we ignore the colony formation units, which of course you have to take, based on what we found, there is definitely something else in colostrum that has a big influence on how the calves uptake antibodies. We're talking about maybe cytokines, complete cells like leukocytes that produce antibodies themselves. We know those things are there, and they are uptaken by the calves. We have molecules like lactoferrin that they are chelating molecules that chelate, let's say, calcium and other molecules that they really have an effect there. So there's definitely more to colostrum than just IgG, but IgG is definitely probably the biggest thing to look out for there.
Yeah, that's true. And well, a practical question now. So if you would propose an index for colostrum improvement or for calf health and failure of passive transfer reduction, would you suggest to work on the dam side or on the calf side? And if calf or dam, and why? So and how difficult will be to record data on a large scale in the population? Yeah,
that is definitely one of the challenges. Translating this study into something that's really applicable. I think something that we were aiming for, hoping for, was that BRICS could work as an indicator trait, which I think it does. The other thing that we wanted to do is to see if we could use that information that's in the colostrum to predict the calf's uptake capacity. But because of the sample size, we couldn't really do it. But what we found is that it seems like it should be possible. Mm-hmm. So, and we're also planning to expand the study a little bit with more samples. But the idea would be to use that colostrum information, the BRICS, to predict the calf's uptake as well. So like a 2-for-1. And now, how to implement this in a realistic way? I think BRICS is, since it's not something you do in a laboratory or anything, simply something you do with a very easy to use machine. I think it's realistic to include that these devices are cheap. And also, since it's colostrum, you don't have to take a blood sample, you don't have to hire a vet or anything. I think it's something that is doable from a breeding perspective. Also, there are some studies going on using infrared information to predict antibodies there. So I think there's a good potential there for application of this in a more realistic breeding program.
Yeah.
But if you ask me, I think there is so much that can be done just by improving management. With just improving management, a lot can be improved. This is something that needs to be tackled as well. And that's something that can be done. No need for anything fancy, just awareness at the farms.
Yeah.
Good.
Laura, are there any other questions from the public?
No, there aren't in the chat, Angela,
Okay.
no. And I think,
Good.
Juan, your final comments there were a nice wrap up and lead in to our next speaker. Thank you very much. And if it's okay with everyone, I can't get feedback from all 119 people in the audience, but we are actually going to adjust the schedule a little bit and go straight to Professor Nina van Keyserlingk, who's up very early in the morning at the other side of the world. And we're going to go straight to her talk and skip the coffee break. So we won't know, and we won't mind if some of you do nip out for a quick coffee, because we won't be able to tell. But I'd just like to say we're very grateful to Nina for joining us. It's a very early start for her. And I think you'll all appreciate, and I think it'll be very evident in her talk, her love for animals, which isn't surprising given that she grew up in a ranch in British Columbia and Canada, and went on to study agricultural science. Her undergraduate degree was in the University of British Columbia, master's degree in Alberta, and then came back to British Columbia for her PhD. So I think, Nina, you worked in agribusiness and industry for a while before coming back to the University of British Columbia as a faculty member in the animal welfare program in 2002. Nina is recognized internationally for her work on dairy cow and calf health and welfare, behavior health and welfare. And I think particularly in recent years for combining qualitative work in this area with the quantitative work. And she's kind of a leader in her field in that area and has over 280 publications, probably more, across all these fields. So very looking forward to your talk, Nina. Thank you very much.
Thank you, Laura, and thank you to you and the others for asking. And I've already had one cup of coffee, so I'm somewhat awake. So I just heard the tail end of John's talk and Juan's talk as well. And so I'm going to riff a little bit off of some of the things that they said. And particularly Juan's last statement is about how much management matters. And that's really what I'm going to focus on today. Let me just share my screen here, if I can. Hold one sec. Does that look good?
Perfect.
Laura?
Yes.
Perfect. Okay. So I'm just going to make myself non-existent here. Okay, so what I want to talk to you about for sort of the next 20, 25 minutes is how calf welfare and performance can walk hand in hand into the future. And there is no doubt, I think it was in John's talk that they talked about the role of assurance programs, and there is absolutely no doubt that questions are being asked about how we care for animals on farm. And historically, animal agriculture has really benefited from this trust without questions from society, that farmers were revered, that they were stewards of animal care, and that whatever they did, the public society trusted that they were doing the right thing. And that whole paradigm is now being questioned. And we're seeing, you know, in Europe, the European Union is got the Animal Welfare Directive. And even here in North America, we're having increased requirements and discussions around the whole idea of assurance and actually providing evidence that you're doing so. So I just want to start where at the very beginning, and I've been working in cows for over 20 years. And, you know, when I first started, and even just as a kid, I was in this farmer 4-H clubs, and I was on the beef side, but I had friends on the dairy, and this was common practice that you saw on dairy farms, is that calves were fed twice a day. They were fed using a bucket, and we provided about 4 liters of milk a day. But a lot of what we do in animal welfare science is look to nature, what would happen in a more naturalistic situation, and not to say that we need to duplicate that, but what are those behaviors that those animals are highly motivated to engage in, and can we duplicate those on the farm side? So when we look to nature, what does a calf do? It nurses 5 to 10 times a day. Each nursing bout lasts about 5 to 10 minutes. So we're looking at about 45 to 60 minutes of nursing every day. And on average, these calves are drinking about 10 liters a day of milk per day. So we are very, very far removed from what nature does. Early in the 2000s, Mike Appleby visited us and did some work where he actually sat with stopwatches and looked at, what happens when we feed these calves. So these were calves that were fed these limited amounts of milk. And what he found is that during each feeding, calves spend about 44 seconds drinking the milk. They just go and they drink it all up. And then, and I'm sure many of you have seen this, what the calf does for the next 6 minutes is suck on anything that it can suck on, be it the chain, the bucket, the pen hardware, whatever. And so our solution to that was not to think about why do calves suck like this, but was to individually house. However, when you give calves access to lots of milk through a nipple, what happens is they spend about 47 minutes a day drinking from that nipple, and they typically spread this into feeding time into about 6 to 10 meals a day, which is exactly how they would spend their time if they were nursing from the dam. So we've also know that, and it's still, I've been traveling. I just came back from Australia, and there's still lots of farms that are feeding 4 or 6 liters a day, and I know that there's still parts of Europe that are still doing this, and I'm really proud to say that in Canada now, as of April 1st, 2023, that the dairy industry themselves have said that it is now mandatory that all calves get fed at least 20% of body weight. So for a Holstein calf that was born, that's going to be about 8 liters a day. So we know that these calves need more milk, but the evidence is also very clear. So Andrea Vieira did some work where she had calves being fed from an automatic feeder. Half of them the feeder was programmed, they can go whenever they want, drink as much as they want. And the other half were programmed so they could only get 4 liters per day, 2 in the morning and 2 in the afternoon. And this is now time spent in the feeder, minutes per day. And what you can see here is that, again, when they have access to as much milk as they want, they spend about 50 to 60% of their time in the feeder. However, when they're fed only 4 liters a day, what you see now is that they're spending almost twice as much time in the feeder. And we predicted that that is evidence of hunger. What Andrea then did is she went back and she looked to see every time they went into the feeder, was it a nutritive visit? So did they actually get milk, or was it a non-nutritive milk visit? Meaning that, especially for the 4-liter calves, their time to get the next allotment of milk wasn't there yet. So they would go in and it wouldn't give them any milk. And so she divided these visits into rewarded and unrewarded. And you can see that those calves that had access to lots of milk, almost all of the visits were rewarded. On occasion, they went in and probably just sniffed the nipple and backed out again. But look at what happened to those calves that only had 4 liters a day. They did go twice a day to get their 2 liters in the morning and the afternoon, but then they went almost 25 times more to get access to look, is it my time now to get milk? And when we published this in 2008, was really the first evidence that these calves fed these low rations were actually experiencing hunger. So Mohammed Khan was a postdoc with us. He did some of this work. He has done a series of papers looking at feeding high levels of milk. And this is some work that he actually did in Korea that I think was really very elegantly done. So these are calves. He had 2 groups of calves. The red ones the bottom line are those calves that are fed conventional, so 10% of body weight. And these were Holstein calves, so about 40 kilos when they were born. And you see just a tiny bit of an increase in body weight because they're not being fed very much. And then he fed them out 10% of body weight until they were about 44 days of age. And then over the course of 5 days, he weaned them, and he weaned them at day 52. The other treatment, he gave them 20% of body weight. And so just over 8 liters of milk a day, increases slightly because they're also gaining weight. And then at about 4 weeks of age, he ramped them down slowly over the course of a week to 10% of body weight, took them out, and then the same as the other ones, he then weaned them over the course of a week and finished weaning on day 52. So this is milk intake, okay? So of course, those calves that are fed 20% of body weight were consuming much more milk, but they were also gaining more weight, and I'll show you that evidence in a second. And so even when they were dropped down to 10%, they were getting slightly more milk than those calves that were conventionally fed. This is starter intake. And one of the beliefs is we don't feed a lot of starter when they're babies, less than 4 weeks of age, because we want them to eat starter. So calves are physiologically just not able to eat starter until they're about 3 to 4 weeks of age. And this is shown here, like they nibble at it. Doesn't matter how much you feed them. There's no difference in starter intake between high-fed calves and low-fed calves. However, remember those calves that were fed with the step down, those that at 4 weeks of age were brought down to 10% of body weight. All of a sudden, we see this dramatic increase in starter intake. They were weaned at day 52, and then Khan took them out to 90 days. And you can see there's a tremendous growth advantage, starter intake advantage of those calves that had been fed lots of milk in the first 4 weeks of life. And this was also realized in body weight gain. So these calves, all started out at about 40 kilos, and look at, we've got almost sort of a 15 kilo difference by 90 days of age. And so what we're saying, or what we believe now, the way to feed calves is give them as much milk as they can in the first 4 weeks of life, because that's what these calves were meant to do, was to take milk and convert it to lean tissue growth. At about 4 weeks, when their digestive system is getting rolling, you can start bringing the milk down, and what you'll see is this increase in starter intake. There's lots of evidence now, and these are just 3 studies, that the more nutrition you give these calves in the first 3 to 4 weeks of life, the more she will thank you in increased milk production, especially in the first lactation. And so this has been done across 3 different studies, and the evidence is now very, very clear. We also have been doing some work over the years looking at the role of starter. And I think because we were feeding these calves so little milk, because we wanted them to eat starter, early days when forage was in the pen, what you saw was that these calves went crazy over the forage to the point where they got these massive hay bellies, and this was not good. And so our reaction was, oh, well, let's just take the hay away, rather than asking, why are they eating all of this hay? And what we found is that 2 things. One, these calves are born as monogastric and they really want to become ruminants. And 2 is, I think eating all of that forage was another sign that they were hungry. They just didn't have the digestive system to cope with it. The issue with hay is, and this is my background also in ruminant nutrition, we always think about things, if they eat more, that must be better. I think with hay, it's not about eating more, it's about eating a little bit at the right time to help transition them from being this monogastric to a ruminant. So here, Khan had 2 treatments. He had calves that were fed 8 liters of milk for sort of 20% of body weight. They were stepped down at about 4 weeks of age, and then they were weaned at 50 or 60 days. Half the calves only got starter during the milk feeding period, and the other calves got starter and hay. And so this is calf age in week and the amount of intake, and I've separated it out by the 2 treatments. And what you can see is that as milk got stepped down at about 4 weeks of age, you see the starter intake increase, but you also see the hay intake increase, but it's very slowly. What we then did is we also had male calves in each treatment, and we slaughtered the male calves, and we looked at rumen function basically. And so here we show rumen pH. So calves fed forage before weaning had larger rumens. So after we slaughtered them, took the rumens out, we emptied them out, we looked at the pH, and we looked at the weight of the rumen apparatus as a proxy for development. And what you can see here is that those calves that had starter plus forage, first of all, had significantly more developed rumens, and they also had higher rumen pH. And so this tells us that having access to some starter, to hay, seems to be positive for rumen development. But remember, these calves were not hungry because they were all being fed at least 8 liters of milk a day. So this amount of forage I don't think it's really for nutrition per se, but they need access to this scratch factor to help them become ruminants. So we started with this limit feeding, and we started with single housing. I think because we were so stressed about cross sucking. And now we know that if we can increase a feed through a nipple, feed them lots of milk, that what we've been able to do is revisit this idea of social housing. So this is a study where we looked at individual housing and pair housing. So these calves are housed exactly the same way, except for in the middle of the pen, we just removed the divider. So they had per calf the same space. They had access to a milk bottle twice a day. They had water. They had starter grain. They had some forage available to them. Andrea Vieira was the PhD student that fed these calves. She fed them out. At 4 weeks of age, she stepped them down, and then starting at about day 42, she started weaning them. And weaned them over the course of, or 48, she weaned them over the course of about 5 days. So slowly ramped down the milk. She always weaned one single calf and a pair house calf at the same time. These calves were all in the same barn. It was single, pair, and then across the alley was pair, single, pair, single. So the singles could not touch another calf, but they could see calves across the alley. So this is the number of vocalizations. And what you can see is that weaning distress is reduced when calves are pair housed. Even though they're all in the same pen and those single house calves could see other calves, it seemed that if you're going to go through a stressful event in life, do so with a friend. Doing so with a friend greatly reduces the distress response. She then took these individual and pair house calves, and they spent another week in those pens. And then she regrouped them into a group house pen. And in that group house pen were 3 calves that already knew how to use the grain feeder, which is here on the left. And they also knew how to use the water bin because they had to put their head in through this thing. And they knew how to use all these things. So she put a pair and an individual. And remember, they'd only ever eaten out of a bucket before or a nipple feeder. And now the milk was gone. So they'd only ever eaten starter out of a bucket. And here, what she did is she looked at how long did it take these calves to learn how to find the grain feeder and have their first grain meal. Time to start feeding. Their first grain meal after mixing was much more rapid when calves were previously pair housed. The pair houses took on average 9 hours to learn to where the grain feeder was. The individually housed calves took almost 50 hours. This was hugely shocking for us because we did all this by a video. We now never leave the calves to learn how to do this by themselves. We always show them. But you can see it was huge. Not surprising, this is the solid feed intake, kilograms per day, and this is days after introduction into the pen with these 3 calves that they'd never seen before. And this is starter intake. And remember, those individually housed calves didn't eat for 2 days. And then I think what happened is that they found this grain feeder and then they ate like crazy. And then I think they got we didn't do this, but I bet you if you'd followed rumen pH, we would have seen some indications of rumen acidosis. Then they dropped, and then they ate like crazy, and then they dropped. The pair house calves never really missed a beat. And so they found the feeder in about 9 hours, started eating grain, and literally after about a week, were consistently eating between about 3 kilos a day. Not surprising, those calves that didn't eat for 2 days, so the red line here, they lost over 2.5 kilos of weight as after being mixed. And then, because I think they gorged on grain when they found it, we see this massive increase in weight gain, drop in weight gain, increase in weight gain, drop. We call this, from my beef days, this is what we call yo-yoing, this increase, decrease, increase, decrease. And I think this is just a function of them trying to cope with rumen acidosis. For ruminants, the best thing, if they've got a tummy ache because they've got rumen acidosis, the best thing for them is not to eat, because then rumen pH goes up again. And in this, we're seeing this in these calves, and when they don't eat, they lose weight, then they get really hungry, and then they gain weight again. One thing that we were trying to understand is how to explain what happened with these, why these individually housed calves took so long to find the feeder. And this is another study done by Andrea Vieira, where she took 2 calves that had never seen each other before. The black calf is a pair housed calf, and the the one with more white, is an individually housed calf. And what you see here is that this individually housed calf is showing much more signs of fear. It took much longer to touch the other calf. And then once it touched the calf, it would never leave it alone. It had 0 social graces. These are 2 pair house calves that have never seen each other. And as I've often said, for those that have heard me present this data, this is a really boring video. Pair house calves that have never seen each other, they entered the arena, they nose touched. And then what they did is they explored the environment. And I often say, if we could have interviewed those individually housed calves in the study after they went into the arena and said, look, tell me about the arena, the individually housed calves never could have done so, because they were so focused on these other calf, and they were like rubber balls, likely explaining why these calves took 50 hours to find the feeder, because they just simply couldn't be calm enough to actually watch what's going on and explore their environment. Since then, we've done some other work where we looked at how socially motivated calves are for social housing. And so this is Thomas Eddy's work, where we had calves that were individually housed in the middle, and they could, on the left side was, or the right, because we balanced for side, where there was an empty pen versus another calf. And here's a picture where we had a push gate, and then every day we increased the weight in the bucket. So calves actually learned how to do this without any training. We did this when they were 3 weeks of age, and then they could then push either for the empty side or the social side. And what you can see here is that from all of these calves, with the exception of the first calf, all calves either pushed nothing for the empty, or they pushed far more weight to gain access for the social partner than they did for the empty pen. So this gives us really strong evidence that they're highly motivated for to have a social partner. I just want to end now with a little bit about benchmarking. The word wasn't, I don't think, brought up in John's talk, but we were talking about how to motivate change. So we've done some studies looking at the value of benchmarking, and this is where we go on farm and we provide farmers with their own evidence, so data from their own farm, but we put it into context of farms in their region. Obviously, all the other farms it's all confidential. They just know that there's these farms that are on their benchmarking report are from the same region. They don't know who they are. And this is where we did this work where we looked at biological outcomes, and then we also did interview-based studies to try to find out sort of what are the most important things that the farmer is looking for, or in this case, we also interviewed veterinarians. And so I've just got some quotes here. This was a farmer that participated in the study. He says, "The value of benchmarking. It gives you a real honest opinion of where you're at. Everyone who I've ever talked to has ever benchmarked is always lower than what they think." I'm just going to say it. Just tell me where I am and tell me how I can do better. I don't really care if I'm at the top. Tell me how I can do better. So in this case, Dax Atkinson, who's now a veterinarian, he went on these farms and we did 2 outcomes. We did colostrum, we measured IgG status, and we also measured calf weight gains. And I'm just going to focus on the calf weight gains here. And for 8 weeks, went every week and did body tape measures and got an estimate of average daily gain from the calves on that farm. And then the farmers got a report. And so they were showed where they were relative to everybody else. And so this is your farm versus the lowest versus the highest. And then what we did is we went and we sat around the milk house table. We gave them their report and then asked them if they wanted to stay in for another 8 weeks in the study. There was one farm that I was at where during the conversation before we'd actually seen the report, and I'm going to name my farmer Tom. Tom said to me, Nina, I'm done. I don't want any of these students running around. My calf feeding program is great. And then he saw the report and he turned to me and he goes, we're in, because he was a great example who thought he was doing everything really well, thought he was one of the best in the industry. And he actually realized that he was the second from the bottom. So so we had these 18 farms that participated in this study, and when they got their benchmarking report, they worked with their veterinarian. They could change whatever they want to change, and then we would move again. So some who hadn't been measuring colostrum quality decided to measure it. Some increased their milk allowance. Some stopped doing abrupt weaning. Some started providing forage before weaning. Some increased teat availability. We gave them agency over what they could do. So this is just an example of calf weight gains, and this is the daily gains. Here are 18 farms, and you could see the lowest was about 500 grams today to some that were almost a kilo a day. So there was tremendous variation. They got to change whatever they wanted to change, and then we measured them again. And so you can see here, these are the ones that changed something. Somebody didn't change, but many of them actually got better. Some either had no change or got worse. But remember, most of them, we did colostrum and average daily gains. Some of them all of a sudden focused a lot on colostrum management. And so this comes back to this complexity of management. We have no idea, but we could predict that potentially they focus so much on colostrum that they let a few of the other things slide. But this is the value of benchmarking. The other thing that was super important to this was the value of the veterinarian. And this is a farmer saying, anytime you talk to a veterinarian about things, you sort of get motivated to even better. You become more conscious of it, you pick up stuff. One of the things that the qualitative stuff showed us is that even when we interviewed the veterinarians in this study, many of them were hesitant to talk to the farmers about calf care because they couldn't link it to some sort of economic outcome. However, when we talked to the farmers, they wanted to hear from the veterinarian. They didn't care. They didn't need this economic hook. Their comments reflected around, look, these are my calves. At the end of the day, I'm going to make the decision that I want to make, but I need all of the information to make the best decision. And so one of these things showed that this importance for the vet also to just be open about things. The farmer is going to do what he wants to do and can do, but without access to the right information, I think we're preventing them, increasing the risk that they're not making the best decision. So in conclusion, feed calves more milk via a nipple. I didn't talk about this, but absolutely feed at least twice a day and don't wean abruptly. Calves want to become ruminants. Forage likely plays a role in facilitating this transition. Individual housing makes calves fearful. I didn't talk about this, but we also know it severely impairs cognition and increases weaning distress. Social housing in pairs or small groups provides multiple advantages. Benchmarking can help motivate farm change on farms. Engaging farmers and veterinarians may provide insights into identifying barriers and preventing change. Thank you.
Thank you very much, Nina. That was wonderful. And as a comment in the chat said, wonderful, illuminating speaker. So I would
thank you for that. It makes particularly at 7:41 in the morning.
Yeah, you did well. You did well. Thank you very much. Nina, I just take the liberty of having the first question. I'm not sure, the chat is open, just to remind the audience that you can put your questions in the chat. And we have some time for discussion. So, and our panel as well are very welcome to ask questions, of course, and to get in on the discussion here. But I love the way you said it at the beginning about looking back to nature. I mean, back to basics, if you like. And in a way, it kind of strikes me. I love your research. I love what you do, and it's so compelling. But it's also so obvious, isn't it? You know, like, I mean, it kind of kills me in a way that I love, you know, work in similar area, but, you know, it's not rocket science. I mean, if we set up a hypothesis that if we're going to set up an experiment to show this, we're rarely proven wrong if you base it on the basic biology of the animal, because, you know, I just kind of get frustrated as to where did we go so wrong in such a short space of time, when where we've diverged so hugely from the basic needs, behavioral and biological needs of animals, and then wondered why we have all these problems, like what John outlined in his talk. You know what I mean? It doesn't seem surprising in a way that we are where we are. And I'm not saying it that, yeah, I don't know if I'm saying it that what we can do to learn from all of that, but I just like that you mentioned it, and it seems that a lot of the solutions are grounded in going back to basics and nature without,
Yeah.
as you say, having to replicate it exactly.
Yeah, I mean, I think the field that we're in, Laura, two things have happened. One, the issue of animal husbandry on farms has been around for a millennium, like it's been around forever. And farmers really have tried to do the best they can. However, what also happens is tradition gets in the way. And you do what you do because your father or your mother or whoever showed you how to do this, or your grandpa, and you do what you do. And then you've got these challenges where you've got power challenges within farm. And we've had lots of, like on that benchmarking study, can't tell you how many times when we talk to the sons or the daughters trying to take over the farm, and all of a sudden, you know, well, yeah, I want to do this, but, you know, grandpa's still in charge of the calves and says no. And so I think there's some challenges that we have with that. So that's why I think at the end of the day, we need to, this is as much a people problem that it is an animal problem. And that's why blending the social sciences has been so important. And I think will become increasingly important, is that we understand these cultural barriers. We understand, you know, we help the farmers with their complex human resources issues, and, you know, trying to get them to talk about these things. And, you know, it's been super cool. Like I just in Australia, where I was presenting some of this work, and there was 100 farmers in the room, and a farmer stood up in front of 99 other farmers and said, I can't believe that I've been starving my calves. I had no idea. The fact that she said this in front of 99 other farmers, to me, was super powerful. Yeah. And it's bringing the farmer voice to the table. That's super, super important.
Yeah, fantastic. And I know there are questions. I'm just going to say one more thing. Would you say that the success of your successes with industry in Canada on calf feeding, because you've jumped ahead of a lot of, you know, other countries in terms of acceptance of the level of feeding and all that, is because you've done a lot of work with industry and conveying that marrying of qualitative and quantitative research to industry and telling them this is what you're telling us, or, you know, because it's just keen to know how you achieved it, basically.
Well, I mean, I haven't been 100% successful. I've been successful in some areas. So I think it's for sure, it's helped to have the social sciences, because we've seen, you know, let me back up. Somebody asked me the other day, what is success? Is success that do we measure success by getting 100% of the farmers to convert to feeding 20% of body weight? Or is success that over time, you know, first we had 0%, now we have 5%, and now maybe we have 60%. And now because of the change, we're going to get to 100%, but that took 20 years. Some could say that wasn't successful. Others can say it's hugely successful. And so I think we have to sort of manage these of ourselves, what we expect. I mean, yes, I think having the social sciences and also trying to understand these barriers has helped big time. Where we failed on the Canadian side was social housing.
Yeah.
So, where it will be the industry, like this is not just for those in Europe, because mostly it's from Europe. We don't have any animal welfare laws. These are all industry driven assurance programs. So the industry has said 20% body weight is mandatory. However, we have a lot of calves in hutches, and they couldn't go there. So it became, we have the humane movement at all the table. It became a horse trade of which things. And so they ended up, I think, phasing out tethering. But if you have your calves indoors, you have to figure out how to get to pair housing. But if you're outdoors, you can stay in hutches. So we failed miserably there. But I think it'll just take another 10 years. The evidence is so strong for social housing.
Really is. And there's a nice question, I think, in the chat there, and there's probably more, Angela, if you want to come in, but I can just bring in this one when you talk about social housing. Essie has said this, thank you, this was very interesting, especially the studies about pair housing. Were there any indications of negative health issues when pairing up, for example, diarrhea, which is always a big concern?
So the two things. One, for those of you that have never fed, for those that are still limit feeding, we have got to this, again, tradition. Tradition in limit feeding is that the feces is very dry and quite hard. And we think that is good. Any female in the room that's had a baby knows that you feed babies high volume of milk, that the feces is loose. It doesn't mean that we're suffering from bacterial scours or something like that. So we actually needed a complete shift in how we score feces on calves with high volumes of milk, because it's going to be much more liquid because they're drinking so much more milk. So that's one thing. What we know from our work and from tons of work done in Europe is that size of group matters when it comes to disease. I do not say, now big groups, huge risk for disease transfer. So what we basically, group size, ideally 5 to 8 calves, maximum 10 calves. But the more calves you have in the pen, the more you got to up the management. And so that's why I'm a huge fan of pair housing, because you manage this group. It's easier, especially this transition from somebody that is only individually housed calves. You get all the benefits of social housing with pairs, but it's much easier to train your workers or to get used to it. To going from individual to groups of 30 is a disaster. So I see somebody asked me, sorry, I just, because it's on this feeding, about, is there room for restricted feeding? No, done. And there is no such thing as feeding too much milk, because the thing is these calves, I'm not an advocate of force feeding calves. So if you're feeding 8 liters of milk and you decide that, oh, the calf only drank 6, I'm going to tube feed it the rest, disaster. So just let them self-regulate. In that first week, you're going to see lots of bouncing, as they get used to the nipple and get things going. 8 liters is the minimum. Most Holstein calves will easily consume 10 liters a day, but you're doing all of this and in the first 4 weeks of life. Then you can slowly, don't do anything abruptly, then you can slowly bring it down, and then you'll see starter take off. But we know that if you've got these increased nutrition in these first 4 weeks of life, Sandra Godden did this in the University of Minnesota, you got improved health outcomes. One of the biggest challenges for any farmer is to get these calves through the milk feeding stage, because that's when they're at increased risk of disease. But that's also because we've been starving them. They've got to mount their passive immunity from the colostrum, as Juan was talking about, and they've got to mount active immune response. That takes nutrition. And if they've got so little nutrition that they can just barely stay alive, no wonder they get sick.
Yeah. There's a big part of the puzzle. There's a question about nipple feeding, which I think is nice as well, about because I think a lot of people still bucket feed in Ireland for sure. I sometimes struggle with the cleaning and the nipple bucket daily versus the ease of cleaning buckets. I see many farmers not cleaning the nipple buckets even weekly. What do you think about providing nipples separate from the milk buckets to satisfy sucking behavior and not feeding a microbial cocktail to the calves?
I'm not a big fan of that because I think the bucket feeding is still going to elicit cross sucking.
Yeah.
Yeah. And the thing is, also remember how the calf drinks that milk. It puts its head into the bucket, it sucks it up. And in nature, I don't know if you can see this in video, but you want them to be like this, because with that extension of the neck, you get the esophageal groove doing its thing, and you get the milk going down. It essentially bypasses the rumen into the abomasum. With bucket feeding, and Mike Van Amberg and Cornell showed this, you get a lot of spilling of milk into the rumen, which is a disaster.
Yeah.
So it really comes down to, and don't make the holes in the nipple really big to get the milk feeding faster. You want them to work. You want all that bicarbonate, that saliva coming down. And yeah, it's going to take a bit of time. You got to clean those buckets.
And I'd love to hear from John if he's seen in his necropsy work evidence of this problem with milk in the rumen of calves that die on farm. Another comment, great speech as always. 2 questions. Do you agree in feeding colostrum in more than 1-2 meals? And if calf is not ready to drink soon after birth, is it better to proceed with a chew? In step down weaning, do you think it's better to reduce the total amount of milk, maintaining the same number of meals, or to reduce the number of meals?
So let me answer the last first, because that's the one I remember first. So I'm not a fan of reducing the number of meals. I mean, these are still neonates, even we're still forcing them to wean at a much, much earlier age. And I think just keeping it twice a day feeding and just reducing the amount slowly, but don't do anything fast, okay? Is the better way to do. The colostrum thing, I'm absolutely a fan of trying to get that calf to drink that colostrum by itself first, but you're also running against a wall, right? And so you need to get the first meal in ideally within the first 4 to 6 hours. Because if you don't, then you've got other problems down the road. So in those cases, as long as the person's highly trained to how to gavage a calf, I can see the merit in doing that, but not as standard practice. There's a lot of calves that are born that are going to suck that colostrum by themselves.
Great. Thanks.
So there is an interesting question about how about using some toys for play for calves kept for individual housing?
So that's about environmental enrichment. I was just at a PhD thesis yesterday about it. So this comes down to one of the things that calves play best with other calves. Calves never evolved to play with a round ball or an umbrella or whatever, right? So what we know, I think we're really at a point where we're trying to figure out what are those behavioral needs that they need that are absolutely mandatory. And so I very rarely use the word enrichment. What I know is that these calves are orally highly motivated. And so we've actually seen the best engagement with something is, if you guys have got old brooms hanging around with stiff bristles, take the end of the broom off and screw it to the edge of the pen. And what you're going to see is that these calves are going to spend a lot of time mouthing these things. Of course, you're going to have to spray them off occasionally and clean them, but they're looking for stuff to engage with their mouth, because remember, in nature, they would be sucking from the cow and they would start engaging in this foraging behavior. They may not eat a lot, but they're going to start exploring. Having a ball in the pen is not going to help with that. So think about things that are more naturalistic. The brushes work well from oral behavior. We actually saw them use it with their mouth a lot more than we thought than scratching. So these are things that I think these are behavioral needs that we need to figure out how to give these calves be able to give these calves the thing to do. Having another calf, they will play as long as they have space. And to me, that is how you elicit play behavior.
Lovely. Yep, makes sense.
Thank you.
Do you have another question?
Yeah, there is maybe a last question. Are there any economic impact studies that show that the money you spend to provide more milk will compensate economically less mortality, less disease, more growth, or whatever?
Yeah, so there has been some work done. I think Alex Bach did some work, and basically, number one, economically, it's going to benefit you downstream with this. The biggest thing is you're going to get this increase in milk yield. So she's going to pay you back down the road. B, Sandra Godden's work showing it less disease, so less antimicrobials having to be used. These calves are healthier, all of these things, and definitely you got growth rates. So, I mean, of course, the further downstream you look, the harder it is to show cause and effect. But I think the evidence is really clear on the health side and on this milk yield side that they're going to pay you back. The other thing is, and for those, when it comes to feeding calves milk, many farmers do increase their milk volumes, but they don't do it in the first 4 weeks. Calves get bigger and they think, oh, okay, the calf should have more milk. And so if you look at the total milk given over the 8 or 10 or 12 weeks, it actually is a substantial amount of milk that they're giving, even though they may have only started at 4 or 5 liters in the first 3 or 4 weeks because the calves are little. What we see is take that total milk and flip it on its head and give 60 or 70% of it in the first 4 weeks.
Makes sense.
So from an economic perspective, it's not such a big of a hit if you look at it that way.
Yeah, but the issue is, are we ready for that? Who is ready for that?
What do you mean? I don't think this is an issue of being ready. I think this is simply the thing that we've got to do. If you're going to be, and this is where it comes back to, the public and the consumer expects animal agriculture to take full responsibility for duty of care. And if they hear, and John probably said, if they hear, and this is the North American data, and I don't think it's that much different, that on average, 7 or 8% of calves are dying in the milk feeding period, they're going to have a heart attack. What are you doing? Whereas, I mean, you look to the beef industry where I grew up, as soon as we've got higher than 3% mortality, the farmers are screaming because they're going to go broke. We in the dairy industry have accepted these high mortality rates as the normal. And that's not okay, because we are actually compromising our social license to practice. Sorry to be.
No, you got to say it, Nina. It's got to be said. It's got to be said. It's got to be said.
Yeah.
And yeah, I mean, a great impactful message to leave us with, really. It'd be great if everyone would just turn on their cameras, Juan and John, if you're still there, and don't put you on the spot so Angela and I can clap for you all. Thank you. No, great way to end, Nina. And on my behalf, thanks a million to all of you for the great webinar. And there's been nice comments in the chat. Congratulating you all and thanking you as well. So really, really appreciate it. And from my behalf, and I'll hand over to Angela to wrap up. Thank you.
Yeah, and thank you to the more than 120 people connected until now, basically. So here you have the link to the Juan thesis that someone requested, and a reminder for the session that will be on the August 30 in Lyon. And the next webinar for sure will be on the 9th of May on poultry science, innovation, and future perspectives. So you are more than welcome to join also the next webinar. Thank you. Thank you, Nina. Thank you, Juan and John.
Thanks a million. Bye-bye, everyone, and thanks to the audience. Bye.